Hello, everybody. All right, so we're going to continue today with phase one of glycolysis. So we're actually going to see some enzymes for the first time, and we'll name them and do a specific thing. So phase one can really be broken down in three. Let me just again move myself a little bit. Steps, if you wish. Although there are 5 steps, 5 reactions really. But in the first, we have a phosphorylation. So we have another phosphorylation later. So where something is going to get phosphorylated, in this case, glucose. Then we have also isomerizations, okay, at 2 positions here, at reaction 2 and reaction 5. And where we just rearrange, let's say, oxygens in this case for the most part. And we also have a cleavage reaction, a carbon-carbon cleavage reaction. This is reaction 4, where we have a 6-carbon unit, fructose, in this case, broken up in 2 3-carbon units. And that will conclude phase 1. Well, actually, what concludes phase 1 will be the isomerization here after that. All right, first one. So phosphorylation of glucose. So glucose comes into cells. We discussed last time how it comes in. It's passive diffusion. So, usually there is a lot more glucose outside from the inside. So glucose will just want to come in. There is a carrier protein that facilitates the transfer. So it's facilitated diffusion. And it comes in. And then once it comes in, the kinase picks it up. So this is a cyclical form of glucose, carbon 1, 2, 3, all the way to 6. And in this case, the kinase is called hexokinase. It phosphorylates 6-carbon sugars, okay? Hexokinase. It moves the gamma phosphoryl group of ATP, okay, onto the 6-carbon of glucose right here, actually on the oxygen, on the oxygen of the hydroxyl group on the 6-carbon of glucose. And as a result, you have glucose that is phosphorylated at the 6th position. It's got a phosphate now at the 6th position. So what kind of reaction is this in terms of the 5 major groups? It's a group transfer. Okay, it's a phosphoryl transfer, and as we had discussed earlier, the group is a gamma phosphoryl group of ATP. Okay, and that is the one that gets transferred. And in general, kinases, all kinds of kinases will transfer this group, the gamma phosphoryl group of ATP, to some acceptor. It so happens that the acceptor here is glucose. How does it happen? So you can imagine R prime X here being anything, as I said, and a kinase, well, what it will do is it will facilitate the nucleophilic attack of X here, these are the 2 electrons, onto the phosphorus of the gamma phosphoryl group of ATP, okay? And one of the oxygens now will leave. All these arrows, the extra arrows here that you see, that it can bounce. The more ways you have to bounce the electrons as they leave, okay, the more you facilitate the reaction. So as a result, ATP now will become ADP. This part is gone, okay? And the R prime X here, whatever that was, in this case, it was glucose, where the X was the 6th hydroxyl group the hydroxyl group on carbon 6. And that now Rx will become phosphorylated. So that's the phosphoryl group, 1 phosphate and 3 oxygens that left ATP. Okay. Depending on what the R prime is, you can have lipids, proteins, sugars in this case, a lot of things, right? Why? Why do we do that? Okay, remember now that gets back to transport. Okay, so glucose is not very charged, and it can come out. In fact, the carrier protein, once it comes in, and let's say there is a lot of glucose outside, and it wants to come in along its concentration gradient, facilitated by the carrier protein. At some point, you're going to hit enough of a concentration inside the cell. Now it wants to come out. If you're a cell and you have something so valuable, nice sweet tasting glucose. Okay, do you want to let it go? Obviously not, right? So what a better way? Uh, there is no better way to from keeping it going outside other than, let's say, phosphorylating it, sticking a very highly negatively charged group on it. Look at that here, okay? And now glucose cannot get out anymore. Furthermore, because glucose 6-phosphate is different than glucose, okay, uh, concentration-wise, with the carrier protein and everything, it's as if there is zero glucose inside the cell. So glucose keeps coming in. The hexokinase keeps phosphorylating it, and everything stays inside the cell. Okay, so that's why you want to phosphorylate. You want to keep it inside, and the reaction decreases permeability, and free glucose keeps coming in from the outside. There are many kinds of hexokinases. They come in many different flavors. We call them isozyme, of course. And hexokinase can act on other hexoses, not just glucose, as the name implies. So it's not super specific for glucose. It will work on fructose, on mannose. They're all cousins of glucose. There are other 6-carbon carbohydrates. So yeast has 3 hexokinases. Animals have 4. And the type 4 is actually kind of special. It's found in the pancreas, in the liver, in the brain, and our intestines. And we call it glucokinase. So it's a little different. Unlike hexokinases, which, as most enzymes do, eventually, you build up a lot of product, and the product will inhibit the enzyme. The reason is very obvious. Both the reactant and the product are normally found in the active site, right, of every enzyme. And eventually, when you have so much product around, I mean, there's not going to be, it's going to occupy the active site, and it's not going to be too much chance, let's say, for the reactant to come in and do the reaction. So that's and proceed, from reactor to product. So that's one reason. Okay, there is product inhibition. However, in most cases, but not in all cases, not in all enzymes. Glucokinase is one of these enzymes that has no product inhibition. Okay. So what happens now with animal glucokinase is that it's very specific for glucose, but it binds weakly to it. It doesn't bind anything else, just bind glucose, no other hexos, for example, but weakly. And it is not inhibited by its product, so no product inhibition. So as long there is, tons of glucose on the outside and accesses it, it will convert it to glucose 6-phosphate. It will phosphorylate it. And because the affinity is very low or lower, okay, for glucose, so it has high Km, it only works really when there is a ton of glucose around, let's say after a very starchy meal, for example. And the levels of glucokinase are positively controlled by insulin. So glucose uptake now is massively amplified. Insulin kicks in, tells you to pick up all the glucose that's found in the bloodstream, and glucokinase is one of the ways that this is achieved. All right, now we're gonna see something that's very common for metabolic enzymes, especially very abundant ones like glycolytic enzymes. Glycolytic enzymes are so abundant that 10 of them that we're going to discuss in glycolysis, just 10 different proteins make up 50% almost of the entire protein composition, let's say fraction of yeast cells. Okay, all the other thousands of proteins make up the other half. Okay, but those 10 enzymes are so abundant that make up almost half of all proteins in a yeast cell, and often they have functions that have nothing to do with the catalytic activity. So this, we call them moonlighting functions, and they do something else. And we're going to see almost for every single one of them, we're going to see some of these moonlighting functions that has nothing to do with what we described as a phosphorylation, let's say, of glucose to glucose 6-phosphate in glycolysis. In this case, hexokinase 2 in yeast actually works as a transcription factor. It goes into the it doesn't phosphorylate anything in the nucleus, but it goes in the nucleus and activates gene expression. So, all right, now we move on to the next reaction. We have glucose 6-phosphate, and we're going to convert it to fructose 6-phosphate. The enzyme is called phosphoglucosa isomerase. Okay. And it's thought to be one of the most boring reactions. It's got one substrate, one product, no cofactors, and it pretty much it works at equilibrium. The delta G is near zero. It is important, so you need it to be able to live on glucose. So, and if you don't have it, you're dead. So yeast on glucose, but you can survive if somebody gives you fructose on the outside in the diet, which is what happens in yeast mutants for this enzyme. So what is the isomerization? It's called a 1,2 hydrogen shift. So what we're going to make really is an aldehyde. We're going to start with an aldehyde, which has a carbonyl group at the very end, okay, of the sugar, and we're going to make a keto alcohol. Okay, that's going to be fructose. So let's see the two products in the open conformation. The ones you were seeing before were in the closed circular conformation. This one's going to be the open conformation. That's glucose, 6 carbons. That's number 6, okay, which has been phosphorylated by hexokinase. How do we know, okay, that, what's the definition of a sugar, by the way? We have to define this. So a sugar is, I'm sure you know this, that's just a review. So from 410, I'm sure you have heard it, but as a review, let's see what is a carbohydrate, what is a sugar in this case. So the carbon atoms is at least 3, and can be higher. In living cells, usually you go up to 7. Some 8 carbon sugars have been described, very unstable, extremely unstable. Up to 7, you find. Glucose has 6 carbons. And in every sugar now, every carbon has an oxygen. Okay? Every carbon. So you don't have anything without an oxygen. No carbon without an oxygen. And one of those oxygens has to be a carbonyl. And all the others have to be a hydroxyl. So you see here in the case of glucose, okay, carbon 2, 3, 4, 5, and 6, they're hydroxyl carbons. Okay, the hydroxyl carbons I have discussed earlier. But there is one that's a carbonyl, and that's the very end. So that's an aldehyde. Glucose is an aldehyde, and we call it an aldose. Okay. Fructose, on the other hand, is exactly the same. It's got 6, in some general properties, it is a sugar. It's got 6 carbons. Every carbon has an oxygen, one of which is a carbonyl. But in this case, it's in position 2, the carbonyl. So it's a keto. So it's not an aldose, it's not an aldehyde, it's a ketone. Fructose is a ketone. So this one is a ketose sugar. So all we're going to do is just really move this carbonyl from the end to from carbon 1 to carbon 2. That's the reaction. So we have, again, the aldose. It's going to become a ketose. How does that work? Well, we have the enzyme that will pick up this proton, and now this bond will make a double bond and push one of the bonds of the carbonyl outwards, and the oxygen will pick up another proton from some general base that happens to be around, and you have this intermediate, where you have now 2 hydroxycarbons, okay, and a double bond. So that's a cis-NADO. It's a key intermediate. It's a signature of this reaction, of this isomerization reaction. Notice that if you look at the redox part of this thing, in ideal intermediate now, these 2 carbons, this is what happened. They became a little more oxidized, okay? And this one, this carbon became more reduced. From a carbonyl carbon became a hydroxycarbon, right? So we do have some internal redox business happening here. But that's an intermediate. What happens next? Then you kind of go in reverse. Some general base, again, will pick up this proton, and now you form a carbonyl here, and you start pushing this double bond out, and picks up another proton, and you end up with a hydroxycarbon. Okay? But at the next one over here, now, where it was a hydroxycarbon, that one became a carbonyl. So what was a hydroxy became a carbonyl, and what was a carbonyl carbon became a hydroxycarbon, right? So we converted an aldose to a ketose through an enediol and intermediate. And again, this is really an internal redox reaction. This carbon got oxidized, hydroxycarbonyl, and carbon 1 became reduced from a keto carbon to a hydroxycarbon. All right, so why bother with this? It generates a sugar with one more primary alcohol. By the way, the primary alcohol will be this. Okay. So, and that's an activatable group, and it's important for that group to be there because a few steps down the road, it will be important to be there so it can activate cleavage between carbon 3 and 4. So to make it a little clearer, let's go back. So you need this primary alcohol up here so you can actually break the bond between 3 and 4 carbons later in the aldolase reaction. So that's the purpose of this reaction. Again, very abundant enzyme, phosphoglucosa isomerase, that is, and it also has a moonlighting role, like hexokinase, which so previously this one of these moonlighting roles is to act as a growth factor, okay? Just out in the circulation, let's say, outside of the cell, extracellular. All right, but back to glycolysis. Now we have a ketose, fructose 6-phosphate. It is phosphorylated at the 6th position. That was the phosphoryl group that hexokinase put on glucose originally. And now we're going to have another kinase reaction. This is the second ATP, okay, we're consuming here. This is the investment part. We're actually spending money before we can make any later, okay, in glycolysis. So what we're going to phosphorylate now is the other terminal carbon, okay, carbon 1. So there it is. So the reaction is now we call it phosphofructokinase because that's a phosphofructose at 6th position, and the kinase recognizes just that sugar and sticks another phosphoryl group at the carbon 1. It's exactly for the same reasons that the same answers that in the what, how, and why for this enzyme apply here. Also, we saw for hexokinase, they apply here too. So it is a group transfer, phosphoryl transfer. How does it work? In exactly the same way as hexokinase. So we're not going to be repeating the same mechanism and everything that we saw before. And why? The permeability constraint. Okay, but you can argue, wait a minute. I mean, it already has the 6 position phosphorylated with negative charges. It will stay in the cell, sure, until it gets broken in 2. And the part that has the phosphate on, carbons 4, 5, and 6, will stay in the cell. But the other part, 1, 2, and 3, will not be phosphorylated if fructose 6-phosphate is clipped in the middle. And you want that too to stay inside the cell. It's valuable. So the permeability from all cleavage products will be, it ensures that there's going to be a block in permeability, so both cleavage products will stay in the cell. So that's why we need to phosphorylate the other end as well. Phosphofructokinase, it turns out, is one of the most studied enzymes because it shows some behavior that's special. So this is the reaction, PFK for short. So we start with fructose 6-phosphate, and it uses ATP as a substrate. Okay, in this case, of course, to do the phosphorylate transfer to generate fructose 1,6-bisphosphate. However, it also uses ATP in a different way, not as a substrate. In fact, that other molecule of ATP binds somewhere else, not in the active site, binds somewhere else on the enzyme, okay, and has an interesting effect. It has a lower affinity. So that ATP binds to the other site with lower affinity, but it does change the activity of, okay, so this ATP binds to a low affinity site somewhere in the protein, not the active site, and that binding affects the catalytic activity of PFK, using the other ATP molecule. And instead of typical, Michaelis-Menten behavior that you see here, where you look at the velocity of the enzymatic reaction as a function of the concentration of the fructose 6-phosphate, that's a typical Michaelis-Menten behavior. But here we have a sigmoidal behavior at the high ATP concentrations, which is a telltale sign of an allosteric enzyme. Okay, So at high ATP, you get this behavior, the sigmoidal behavior. So it is a model of allosteric control. And what does that mean again? It means that the enzyme activity can be regulated by an NF vector that interacts with the enzyme at some site other than the substrate binding site. Okay. So the problem here is that okay, people study this. It shows this behavior in vitro, and if you look at it at different concentrations of ATP, low ATP will have the Michaelis-Menten type behavior, but the high ATP you have this green sigmoidal behavior. But the reality is that inside the cell, as we have discussed already, ATP concentration doesn't change that much. Okay, so this mode of regulation, it's unclear whether it's actually is super biologically relevant. It's something we see in a test tube for sure. But if the levels of ATP don't change much, then what's the physiological significance? So that's an open question. It is a heavily regulated enzyme. In addition to ATP acting as a negative allosteric regulator, there are positive allosteric effectors as activators. So let's go back to the previous slide, and that will make more sense. So basically, the higher the ATP concentration, okay, you will inhibit the behavior of the enzyme, the catalytic activity of the enzyme. But as we said, this may not be so physiologically relevant, but some other effects that actually activate the enzyme, we know now that they are biologically relevant. So AMP, okay, will act as a positive allosteric effector, and fructose 2,6-bisphosphate will also act as another positive effector. So why AMP is more physiologically relevant than ATP's role as allosteric effectors. Here, they both act as allosteric effectors in this scheme, one inhibitory, the other activating. And the idea is that you need ATP to be inhibitory because you have made a ton of ATP. Why would you want to go down glycolysis and break down glucose and get more ATP, right? So that's the whole point. So you block PFK if there is a ton of ATP around, and you don't have to break down your glucose. You just keep it and store it and put it in storage, so to speak. But again, the problem with this model is that ATP levels don't change that much in the cell, so you won't block anything. But if you actually look at the pools of these nucleotides, ATP, ADP, and AMP, so there's a triphosphorylated, bisphosphorylated, and monophosphorylated adenosine. They look like this. Actually, ATP is even more. We've already seen how there is a lot more ATP than ADP in cells. AMP is even lower. Okay? And there is an enzyme that converts these forms. It takes 2 ADPs to make 1 ATP and 1 AMP. It's called adenylate kinase, so it brings back the pools to equilibrium, to these more or less levels, okay, ATP, ADP, and AMP. So the blue stuff, it's an ocean, it doesn't change too much. Okay, so you change a little bit the ATP levels, like a few drops in the ocean. But AMP, on the other hand, a small change in ATP here through reactions, let's say, in the cell, will lead to a huge change in AMP. So in fact, as a sentinel metabolite of the energy status of the cell, okay, AMP is extremely uniquely positioned to do that. Okay, it's very sensitive to changes at ATP levels. ATP, even if it drops by a little bit, maybe 10% or whatever, it won't make much of a difference on ATP levels, but it will make a lot of difference on AMP levels. So the cell senses that, okay, drops a little bit ATP here, AMP will go up, okay, by a lot, not 10%, but several fold. And now that's a signal that's interpretable by the cell. It says, okay, go get me ATP. And the way to go do the ATP is to really help, fructose, to help PFK, phosphofructokinase. It's a positive effector and will drive the reaction forward. Okay, so to make fructose 1,6-bisphosphate. So it's a key control point. And probably the more physiologically relevant effector is AMP and fructose 2,6-bisphosphate, by the way. They're both they activate the enzyme. Remember, with ATP we were somewhere here, or the sigmoidal here with the at high ATP levels, but now you have AMP and fructose 2,6 bisphosphate, and you see a massive activation. It is also one of the most studied enzymes, because when we talk about gluconeogenesis later on, when you have to actually make glucose, not break down as we're discussing here, whatever works in one way to affect PFK, it works in the opposite way to promote dephosphorylation of fructose 1,6 bisphosphate by the enzyme, FPP enzyme shown here. So the enzyme that dephosphorylates fructose 1,6 bisphosphate is called FPP, and will make fructose 6 phosphate, and that's important so we can go back and make glucose. And the effectors that activate PFK, they also inhibit, the enzyme that help you make glucose, which kind of makes sense. Okay, you don't want to break down glucose at the same time promoting the making of more glucose.